8 o'clock, we're recording. Speaking to the people online, I'll remind you for attendance. If you're online and you're watching live, you need to send me the email with the keywords sometime before 9 a.m. So send it in quick. If you miss that opportunity, then you'll just have to answer the questions. And the first word for attendance today is soccer. The other announcements, same ones I've been making. Remind you about lab. The lab for last week was due on Friday. I still haven't gotten around to grading all of them. So as far as I'm concerned, as long as it's turned in before I get around to grading yours, I consider it on time. So if you're thinking, oh crap, I forgot about that, go ahead and turn it in and you'll be all right. And also to remind you, the same thing can be said for the pre-lab. So the pre-lab for this week's lab was due on Sunday, last night. But as long as it's done before I get around to grading it, then as far as I'm concerned, it's on time. So any questions about labs? Yes?
You need to turn the pre-lab in.
Oh, good question. I think if I'm understanding it correctly, she's asking if you need to turn it in, just like everything else. Just always hit the share button. Never hit the submit button. That's what you do. And of course, as always, bring your computer into lab because that's how you're going to turn everything in. But as always, if for some weird reason, I shouldn't say that, I'm sorry. If for some reason you prefer a paper version, just let me know beforehand and I can print one out for you. So yeah, come to lab Tuesday on Wednesday. Make sure you've got closed-toe shoes, long pants. Yeah, and a computer or scrap paper. Any other questions about lab? Okay, next, Google Classroom. Read the announcements. Again, I try not to post too many of them. I'm gonna try to do it like once every few days, but I can tell most of you aren't reading. Don't ask how I know, but I know only a few of you are reading them. Read them, they're there to help you. Especially at the end of the semester, you're gonna be like, some of you are gonna be asking what you can do to fix your grade, and I'm gonna be saying you should have been reading these things all along. Because it's all right there. I'm telling you right now how to get a good grade in this class. Anyways, anybody having problems with Google Classroom? Okay, next, office hours. I just wanted to remind you that on Friday, my office hours are going to be virtual. Not that it seems to matter. Nobody comes to visit me physically in the office anyway. But if you need me on Friday, I'm only going to be online. Speaking of which, speaking of office hours, if you get feedback on anything, right? So now that you've done lab 1, and if I've graded it, you've got your grade back. Maybe you've done the study guide for chapter 1, the study guide for chapter 2, and you want feedback. If you meet me and allow me to give you thorough feedback, you get at least a 5% boost to whatever grade that was. And maybe, depending if I need the video or not, if you let me record it and share it with your classmates, then you'll get a 10% boost to that grade. So for any assignment you ever do in this course, if you visit me during office hours, or arrange a time outside of office hours, and let me give you detailed feedback, you'll just get an automatic boost to your grade. So any questions about all the announcements? All right. Oh yeah, independent work's not up there. Just to remind you, you should be working on your independent work. Please don't wait till the end of the semester. I see it every semester they do. And independent work is sort of like a free 100 points, right? You got to put the work in, but it's free because you're not getting tested on anything and you get to choose the assignment. All right, any questions?
Actually, yes, for chapter 2.
Okay, chapter 2 question.
Okay, so we talked about heat and water, right? And if heat is, if it's being heated, it's faster movement of water, right, and they expand. But cold water also expands. But is the difference the bond's breaking and the bond's not breaking?
Yeah, so almost everything expands when it gets heated. So that part's normal, right? Because what we said is heat, basically molecules move faster. So that part should be easy. Like you said, it sounds like you got it. Water is more difficult because as it gets colder, it gets smaller, but you don't need to know. The part that you do need to know is that once it crystallizes, the bonds aren't broken, but they, instead of moving around like they used to, they're now stuck and they quit moving. Because they're stuck and they quit moving, that's what makes them larger. Just because it's like they're no longer moving around each other. They're just in that frozen position. But a better answer to your question, this is important for the rest of you too. You don't even need to know it at that detail. I taught that detail just so you understood why it happened, to hopefully just help you remember what you do need to know, which is that when ice freezes, it expands. That's the most important thing. You don't need to know why, but again, I'm gladly trying to help you understand why. But just know that when ice freezes, it expands. More importantly, or in this context, the reason that is so important is because the fact that it has expanded makes it less dense than liquid water, therefore it floats. And remember, we talked about why it's so important that ice floats. Good question.
So it causes the blanket to help the
Exactly.
Yeah.
Yeah. Yeah, keep everything else from freezing. The ice is a blanket, as odd as it sounds. It is relatively warmer sometimes than the colder air on top of it. Any other questions? Good questions. All right, this brings us to chapter 3. And speaking of which, if things go as planned, we will finish chapter 3 this week, which means we will have the first exam next week. So go ahead and start thinking about when you want to have the exam next week, whether you want to have it on Monday or Wednesday or Friday. Because once we get a little bit closer, I'll send out a message and ask you guys what your opinion is. And generally speaking, it'll be like a vote. So whichever one gets the most votes wins. Anyway, so obviously chapter 1 we finished, right? That was just like a preview of what the rest of the semester was about. That was kind of defining, if you will, life, defining science. And then we moved on to chapter 2, which was chemistry. And chapter 3 is a little bit like chemistry. Remember chapter 2, when we talk about chemistry, we learned a lot about molecules and bonds, right? And the big four elements, carbon, hydrogen, oxygen, nitrogen. In this chapter, we're still kind of talking about chemistry, but now we're going to be more specific. We're going to talk about the molecules of life. So now we're not going to be talking about any old molecules. We're going to be talking about some very specific molecules, the molecules of life. Every chapter starts with this, why this chapter matters. So in this question, the question of this chapter, the question is, why do biological molecules matter? Any idea? This is kind of hard to see, but can you tell what she's holding? It's hard to tell. She's holding a steak. So then the question that your book poses, and if you read it, I guess don't answer it, but the question your book poses is, what's her hair and that steak have in common? If you're thinking about biological molecules, even though I haven't talked about biological molecules yet, let's say you can guess. Can you think of a certain type of molecule that hair and steak might have in common? No guesses? It's all right. Protein. Protein is what those have in common. So meat, hair, fingernails, a bunch of other stuff you're going to learn about. Protein is the thing that's in common there. So here we go. Got a picture of a kid eating a piece of chocolate. What's happening to that piece of chocolate? Can you tell by its face? Then it's kind of hard to see.
It's melting.
It's melting, right? And I'm sure you guys have seen that before, right? When you're holding chocolate, very often it melts. So why is it that it's solid when it's at room temperature, which isn't that cold, but then it starts melting when you hold it? The answer is, this is something about the way fats are built, so to speak. And you're going to learn about that in this chapter. And then your question, your book mentions the elephants. I'm reading to remind myself of what they're saying. Oh yeah, your book just points out that if you were to look at our DNA or your DNA compared to the elephant's DNA, or even the DNA of that tree, or the DNA of that grass, or the DNA of the worm living in that soil, if you were to look at it, like look at the molecules itself, not the whole molecule, but just zoom in and look at the molecules, they'd be identical. It's one of 4 bases that you're going to learn about. It's got the same backbone that you're going to learn about. So that's your preview. Any questions about that? All right. Your book also has this for every chapter, biology and society, and that kind of gives you a theme for the chapter, but we're going to skip through it as we always do. You're going to learn about lactose later in the chapter, but lactose in the grand scheme of this chapter, or in the grand scheme of this exam, or in the grand scheme of this semester, lactose is not that important. We'll talk about it a little bit, but we're going to skip over it. All right. So this chapter is broken down into 2 main bullet points. Again, I can see it's very hard to look at, very hard to see. The first main thing we're going to talk about is organic compounds in general. Then we're going to talk about large biological molecules and look at them individually. So before we get looking at the specific things, I'm going to give you an overview of things you need to know to understand those things. As you already know from the last chapter, and I told you needed to know this, so I'm telling you again, so obviously it's important. Your cell is mostly made of water. Know that. And remember, in the last chapter, they gave you a number, and that number was 70 to 95%. So know that number. And that says a lot. Like, think about that. Even if you go to the lower number, 70%. That means if you were to look at a cell, only 30% of it is stuff other than water, right? The rest of it's water. Speaking of the rest of it consists mainly of carbon-based molecules. So you are mostly water and carbon-based molecules. You don't need to write down this next bullet point if you're note-taking, because it's kind of a preview of what we're going to talk about. But I'll point out that carbon forms large, complex, and diverse molecules, which makes sense because if most of your cell, other than water, is made up of carbon molecules, you've got to imagine you've got all kinds of different molecules in your cells, right? So it makes sense that carbon can form all these different things. But again, no need to write that down yet, because that's just a preview. The next bullet point, if you're a note taker, you might write that down because you do need to know it. Organic compounds are carbon-based molecules. So when you hear in the context of people in biology, when they talk about carbon-based or organic compounds, when they mean organic, they don't mean like the non-pesticide version of the fruits that you get at the grocery store, right? Not the organic section. But organic in this case means carbon-based. So if you want to trick somebody, you can say, you're not giving your kids organic water. It's not carbon-based. Anyway, any questions about this slide? It's more of an introduction. Let's get into carbon chemistry. That's the next bullet point, which again is really hard to read, but yes, carbon chemistry is what we're getting into. Some of this is going to be a review to you. I've said these things before, but now I'm telling you, and I'm also telling you that you need to know them. Let me think about this. Yeah, and I'm sorry, because I think in chapter 2, when I talked about this, I said you didn't need to know this, but that's when I was listing out all those different atoms, and I was saying you don't need to know how many times they bond. But if anything, know how many times carbon bonds, which is 4 times. And we've already talked about why, right? We said its atomic number is 6, which means its proton number is 6, which means it has 6 electrons, which means it has 2 on the inside, and it's only 4 left, so 4 on the outside. We need 8 to be full, so that means it needs 4 more, therefore it can bond 4 times. And I did tell you for chapter 2, that is what is most important, what I just told you. That thought process is what you're going to need to do in the exam. I'll give you an element, give you the atomic number, look up the protons, therefore the electrons, therefore you can figure out how many electrons are on the valence rings and how many times it can bond. Anyway, so if you're taking notes, the only thing on this slide that I would write down is that carbon bonds 4 times. Yes, it's versatile, but that's what we're about to talk about this whole chapter. Yes, it varies in size. It can vary in branching patterns. I'm going to give you some examples, but there's no need to write that down. Here's just 4 easy examples. These are carbon-based molecules. When I say carbon-based, what I mean is it's kind of like a carbon skeleton. This one has a very short carbon skeleton, very short and straight, right? It's got 2 carbons and just a bunch of hydrogens on the outside. This one's got more of a upside-down T look to it, right? It's short carbon, short little skeleton, and it goes up. And again, just a bunch of hydrogens on the outside. This one's a little bit longer than that one, except it has a double bond, and we're going to learn all about those a little bit in this, a little bit today, and a lot in lab. And this one's completely different, right? It's a circle. Now, how many of you are chemists? None, right? So if I were to ask you how these things behave chemically, like in chemical reactions, could you tell me? No, right? But what you could tell me, just by looking, because I said this in chapter 1, the whole structure function thing, without even knowing much about chemistry, you can just see that they have different shapes, right? And because they have different shapes, they're going to act differently chemically. And that's all you need to know in this course. It's a 100 level course. Just know by the fact that they look so much different, they're going to have different properties. They're going to act different in chemical reactions. Anyway, any questions about this slide? Obviously, those are just examples. You don't need to know those. Here's something you do need to know, and you've already wrote it down, hopefully, in the last chapter. The carbon mostly bonds with these other elements, hydrogen, oxygen, and nitrogen. Because again, this is the same information I gave in the last chapter, except the way I presented it was big 4, right? 96% of your body weight is comprised of carbon, hydrogen, oxygen, and nitrogen. There it is again, presented in a different way. But if you want to look for independent work, that's why I put this little star here as a little reminder to remind you, what are some of the other things commonly bonded that it commonly bonds to in your body? Or I guess if you're interested, you can even look this one time outside of biology. What else does carbon commonly bond to outside of life. Anyway, any questions about this slide? Just a review, right? Pretty simple. And even though I told you to write it down, or suggested you write it down, you almost don't need to. Unless you're just completely zoned out every time you come to class, you're going to know these big four because we're going to talk about them over and over again. And you're going to draw them in lab, unless you already have. What you can do, you can work ahead of slide. Your book gives this example. If you're writing things down, give your writing hand a break because you don't need to know anything about it. It's just an example of a methane, CH4. This is one carbon bonded to 4 hydrogens. It's one of the simplest organic compounds. It's abundant in natural gas, so that's usually what natural gas is, the CH4. It is produced by prokaryotes, which are things like bacteria in swamps. So that's where you get your swamp gas. It's also produced in the digestive tract of grazing animals, so like cows, buffaloes, things like that. That's where it's produced. Just an example, you don't need to know anything about it, but there you go. This next picture is a picture of it, different ways of drawing it. This kind of goes back to something I showed you in chapter 2. I think it was formaldehyde, and the point was there's different ways of drawing atoms, right? You don't need to know them, but I'm just saying, usually I'm going to draw things like this. So for some reason, you see me draw something like this, and then you like to talk about it when you get home, and someone shows you something like this. Don't be like, it's supposed to look like this, right? No, there's just different ways of drawing atoms. That's all I'm showing you. Excuse me, molecules. Any questions about that? And again, I'm not going to like show you this picture on the exam and say, is that a space filling model, a ball and stick model, or a structural formula? I'm not going to do that. All right, some other examples. You don't necessarily need to write this down. Just something to keep in mind as we move forward. The larger organic compounds are the main molecules in gasoline as well. And obviously, this is not a class about petroleum, so you don't need to know that. It's just a good example. So this is what it looks like, right? Octane, that's a large organic compound. That's mostly what you find in gasoline. It's also very similar to the stuff that you find in your body, dietary fat. And again, like I said, you guys aren't chemists, and I showed you those molecules, and I said, do you know how they behave chemically? And you said no, that's okay. But I said, what about structure function? Think about structure function. You can see that these things have similar structures. This dietary fat kind of looks like 3 octanes stuck together, right? It kind of is, which is interesting because, again, similar structures, similar function. The function is to provide fuel. Anyway, again, these are just examples. We will talk about dietary fat later, but right now this is just kind of an introduction. The next slide I'm going to show you is another way of looking at octane and dietary fat. So again, this is just going to be another example. But are there any questions about this slide? All right, the next word for attendance for those of you who are online is going to be cheeseburger. Cheeseburger is the next word. It's right there, it is. All right. So yeah, this next picture, this next slide is going to be the drawing of the actual molecules. So again, you aren't chemists, but here's octane, right? That's what it looks like when you draw it. And here's a saturated fat, right? That's one of the 3 things that are stuck together. You can see how similar they are. And again, structure function, right? They have similar structures, they have similar functions. All right, this next slide is a little bit more important. So the unique properties of all these different compounds, all these different molecules we're talking about, it depends on the carbon skeleton, right? Because like we said, some of them are circular, some of them are long, some of them are branching, some of them aren't. Some of them have double bonds, some of them don't, right? So all those things give them their unique properties, which is why I asked you to say, look at these things. Do you think they act different? Can you tell me how they act differently? You couldn't, but that's fine. All you need to know is the fact that they look different means that they also have unique properties. And it's also not just the carbon skeleton itself that gives it a unique property. It's also the things that are attached to it. So again, if we're talking about the big 4, right? Is it attached to hydrogens? Is it attached to oxygen? Is it attached to nitrogen? All those things make a difference. In this 100 level class, we're not going to get too deep into how much of a difference those things make. The biggest thing you need to understand is the fact that that it bonds to different things. Means it has different functions, unique properties. Which brings us to the next word. So instead of just being attached to other atoms, because again, methane was 1 carbon attached to 4 hydrogen atoms, sometimes the carbon skeleton attaches to functional groups. And you need to know what a functional group is, just so when I use the word, you know what I'm talking about. Probably won't even ask you to define it in the exam, but just in case, a functional group is defined as groups of atoms directly involved in chemical reactions that are attached to the carbon skeleton. Basically, what a functional group is, I think a better way to think of it is, we've got this carbon skeleton, and sometimes it attaches just straight up to an atom. Sometimes it attaches to basically other molecules. I call it other molecules, but obviously once you stick them together, they are one gigantic molecule. But what I mean by that is, you have this other, it's a molecule with a bunch of atoms doing their own unique thing, and then it gets attached to the carbon skeleton. Each of these functional groups plays a role in chemical reactions because they all act differently chemically. And many biological molecules have two or more. So these last two bullet points aren't as important. I would say the most important thing is this bullet point right here. Functional groups are groups of atoms that are directly involved in chemical reactions attached to the carbon skeleton. And also know that all this stuff we're talking about, this bullet point right here, this is where all these unique properties come from. Any questions so far? All right. The next slide I'm going to show you is I don't think it's even from your textbook. I think it's just an example. I'm going to show you all these different functional groups. You don't need to know anything about them, but it shows you what they look like, it shows you the name, and it shows you the properties. So for example, this thing is polar and this thing is nonpolar, meaning this thing is charged, this thing is not charged. And as we know, charged things act differently, right? Positives and negatives come together. Another way you can look at polar and nonpolar is, for example, water is polar and oil is nonpolar. And those things don't mix, right? They stay away from each other. So all these things matter. If you have a molecule that has a polar thing, a bunch of polar side chains, we're saying functional groups, a bunch of polar functional groups, and then another molecule that has a bunch of nonpolar functional groups, you'll probably guess that they're going to stay away from each other. They're going to repel each other. Not that you need to know that. I'm just saying the point here is all these different functional groups have all these different properties, and therefore they interact differently. Which at this point in the game is not that important. But when we start talking about enzymes, a little bit when we start talking about DNA and RNA, all these functional groups are really going to be important. Not that you memorize each one, just that you know, but they have different properties, therefore they interact differently. So anyway, any questions about this slide? All right, move forward. The next thing we're going to talk about, before we really just jump into the meat and potatoes of this, we're going to talk about giant molecules from smaller building blocks. I know that's hard to read. But on a molecular scale, many biological molecules are gigantic. And that earns them the name macromolecules. You don't even know that word. We're not even going to use it. So technically everything we're going to talk about, well, almost everything, is a macromolecule. It's a very large molecule. So it's not like the small ones we learned about when you first learned about covalent bonding and ionic bonding. We're talking about very large molecules. So you don't need to write this down either. But the three categories of macromolecules are carbohydrates, proteins, and nucleic acids. The reason you don't need to write that down is because there's a fourth category of molecules that's important to this chapter. That's the fats. But technically fat is not a macromolecule, therefore it didn't make it to this slide. So your book included this, so did I. But I want you to know you don't necessarily need to know what a macromolecule is. I'm not going to make you distinguish between macromolecules and biological molecules in general. And if you were to look at it like this, abbreviate. I already tell you, we are studying biological molecules in this chapter. Well, biological molecules, there's two types. There's these macromolecules, and then there's the fats. What I'm also telling you is, and that's what this information is giving you, what I'm also telling you is, I'm not going to ask you to distinguish that. But there it is, just in case you want to know. Because again, it's in the textbook, it's a 100 level class, I like to teach out of the textbook. So any questions about this slide? All right, and you'll notice, as we talk about carbs and proteins and nucleic acids, you'll see how much bigger they are compared to the fats. Much bigger. Not that matters. All right, macromolecules are polymers. So again, when we're talking about the proteins and nucleic acids, they are something called polymers. And this is stuff you definitely need to know. You need to know the relationship between polymers and monomers. Basically, monomers make up polymers. Think of like a pearl necklace. The whole thing, the whole pearl necklace is what you would call a polymer. And it is made up of individual pearls, so the pearls would be the monomers. Or a train, right? A train would be the polymer. Each individual train car would be the monomer. And you need to know this because I can tell you this now. Once we move forward and start talking about the proteins, fats, nucleic acids, the carbohydrates, one of the things I'm going to tell you about each of them, I'm going to tell you what the monomer is, and I'm going to tell you what the polymer is, and you're going to need to know that. This portion of the exam is going to be a lot of comparing and contrasting. It'll be like, all right, which of these four molecules has this monomer and this polymer, right? So that's how you're going to distinguish them from each other. One of the ways you're going to know the difference between a DNA, for example, versus an amino acid, a protein. So you can know the name of the monomers and the polymers. Almost sounds like a 60s band. Matter of fact, that would be the next attendance word. The mamas and the papas. Look them up if you don't know them. You don't have to do that. Good. Anyway, polymers are made up of monomers. Pretty simple, right? Here's where it gets a little bit more challenging, but not much. But let's add to that. Now that you know that monomers make up polymers, let's talk about some verbs here. What do you call it when you put some monomers together to make polymer? The answer is a dehydration reaction. It's a lot of words right here. So I don't know. If you're shorthanding, you can just put dehydration reaction equals building. As far as we're concerned, that's just putting stuff together, right? You can even write that. That's the key here. You just need to know that when we're putting stuff together, it's called a dehydration reaction. Pretty much anything we talk about in this semester, whenever we're putting molecules together, it's called a dehydration reaction. Sometimes they get specific and give it its own name, but it's still a dehydration reaction. So all this other stuff, looking at it, yes, it does link two monomers together. That's just a good textbook way of saying it puts stuff together. And yes, technically it does remove a molecule of water. I'm not going to ask that you know that. So I'll put an X there, not because it's not true, but because I'm not going to ask you that. But if it helps you remember, well, there you go, right? Because when you get dehydrated, what happens? You're losing water, right? So if it helps you remember that if you remove a molecule of water when you do that, so be it. The next slide I'm going to show you is a drawing of a dehydration reaction happening. But before I move forward, does anybody have any questions about this slide? Again, for the exam and for understanding everything I'm about to say for the next few chapters, you need to know what a monomer is, you need to know what a polymer is, and you need to know what a dehydration reaction is. And right now, that's kind of easy. The bad news is there's a different word when you break apart something. Right? You ever talk about building something? Well, we have a different word when you break it apart. Cross that bridge when we get there. The thing that stinks is the other word, a lot like this word. It can be a little confusing, but we'll get there. So if there's no questions about this slide, let me show you the picture. Here we go. Here's a short polymer, and we're adding a monomer to it. That is a dehydration reaction, again, because like I said, we're just building stuff, right? Or putting stuff together, or adding a monomer to a polymer. And yep, that's a dehydration reaction, and you've squeezed out a water molecule, so to speak. Any questions about that? All right, obviously this is just an example. They didn't even give these things names. I don't know what kind of polymer this is. I don't know what kind of monomer this is. It doesn't matter. It's a generic polymer being attached to a generic monomer, and that is a dehydration reaction. And another thing they could have put is this could have been a monomer. You could have had a monomer and a monomer. Putting them together, it's still a dehydration reaction because you put something together. Or this could have been a short polymer attached to another short polymer. That would have been another dehydration reaction because you're putting something together. Or it could have been a long polymer and another long polymer, right? The point is, if you're putting stuff together, it's a dehydration reaction. That's what you need to know. So let's talk about the opposite of that, just to confuse things. So right now, that's kind of easy to remember, right? Now you build something, it's called dehydration. Well, when you break it, it's called hydrolysis. To me, that was always confusing because both of the names have something to do with water, right? Dehydration and hydrolysis, and I was confused. But yeah, so know that dehydration is when you're doing it, and hydrolysis is the opposite, it's when you're breaking it. That's really it. I mean, yeah, there's a bunch of other words on that slide, but really that's all you need to remember for this exam and to understand the things we've been talking about this semester. When I say dehydration reaction, I'm talking about building something. I say hydrolysis, I'm talking about breaking it apart. And again, like I said, you don't need to know that dehydration reaction takes the water molecule away. Same thing we said for this. You don't need to know that hydrolysis adds a water molecule. But if it helps you remember that's what it is, then so be it. There's a picture of it happening, right? So this is the opposite of what we just saw. Instead of two things being brought together, we have one long thing, a water molecule kind of splitting it apart and into two shorter things. This time they didn't need to label it. I like it. This is what they showed us showing on the other side. They didn't need to say long polymer and then short polymer and shorter polymer, right? It doesn't matter. The point is, if you're breaking something apart, it's a hydrolysis reaction. So any questions about monomers, polymers, dehydration reactions, or hydrolysis? Very important things for everything we're about to talk about. When we talk about proteins, carbohydrates, fats, and nucleic acids, right? Those four things are the main four things we're about to learn about. The main four things that this chapter is about. This is what you're going to need to know about. You need to know about the dehydration reactions that make hydrolysis reactions that break. What is the monomer of a carbohydrate versus the monomer of a protein? What's the polymer of a carbohydrate versus the polymer of a protein? They're all things you need to know to distinguish between those four things: carbohydrates, proteins, nucleic acids, and fats. Still see a little note taken, so I'm going to slow it down a second. Before we move forward, I guess I'll give you another announcement that I forgot to remind you of, which you probably know by now. But remember, on Wednesdays, if you show up in person and you wear WVSU gear, you get a little bit of extra credit on your attendance. And then if you take the picture with me, you get a little bit more extra credit. And if you were to tag yourself in that picture and or share the picture, you get a little bit more extra credit. Or if you're online, can't come in and take a picture wearing WVSU gear. If you send me a picture of yourself in WVSU gear, then you get a little extra credit. Anyway, I see that note taking is done for this slide, so let's move forward. This is the important part. That first main bullet point was just the background. That's all the background information you need to know to understand this. This is the meat and potatoes. We're going to study carbs, lipids, proteins, and nucleic acids. Most of the exam, for this portion of the exam, is going to be able to differentiate between these four things. Again, what are the monomers of this one called? What are the polymers of this one called? What are the properties of this one? What are the properties of that one? What's this one do? What's that one do? Be a lot of comparing and contrasting. No need to write those down, I think. Because again, we're going to discuss each one of these four separately. And basically, this next bullet point says the same thing. Four categories of large biological molecules that are found in all living creatures are these things: carbs, lipids, proteins, nucleic acids. Of all living creatures, anything that's alive that we know of on Earth have these four things, right? These four molecules. And I chose these pictures. Does anybody know what this is? Called tardigrades, or water bears, is a watcher bear. It's one of their nicknames. It's, I think, the smallest animal on Earth. If you're interested, you should look into them for independent work. They're very interesting. You can radiate them, boil them, put them in space. It's hard to kill them. Anyway, that's the smallest animal on Earth, and this is, depending on who you talk to, the largest tree on Earth. That you're probably looking at and like, that's not a tree, that's a bunch of trees, right? But it's actually one tree. And that's a whole different discussion. But anyway, the point is, whether we're talking about the smallest animal on Earth or the largest tree on Earth, they all have these four things in common. So let's talk about these four things. First one we're going to talk about are carbohydrates. And what I'm going to do is give you a little bit of background information about carbohydrates in general. Then I'm going to tell you about the 3 different types of carbohydrates that you need to know about for the exam, which are monosaccharide. I need to write this down. We're going to talk about them separately. But under the category of carbohydrates, we have monosaccharides, disaccharides, and polysaccharides. So for the exam, first of all, you're going to need to know that these 3 things fall under the category of carbohydrates, but also you need to know the difference between them. How is a monosaccharide different from a disaccharide? How are those things the same? How are they all similar to each other than the fact that they all fall under the category of carbohydrates? Well, let's jump into it. So, surely you guys know a little bit about carbs, not even from a biological standpoint or biology class standpoint, but just from eating, right? So, what do you see here in the context of carbs? Pasta, good, yeah. So if anybody runs, I don't know, I don't do this, I run, but I don't do this. But anyway, I guess serious runners, supposedly before a race, a long run, they'll do carbo loading, right? So they eat a lot of things like pasta. And hopefully by the end of this chapter, you'll understand why. But yeah, pasta's the big one. When people think of carbs, I think very often they think of things like pasta. What else do you usually think of when you think of carbs? What was it?
Potatoes.
Potatoes, yeah, that's a good one. That's actually the one on the highest glycemic index, which is something we don't talk about in this class because it's not a nutrition class. But what else? Pasta, potatoes, anything else? Bread, that's a big one. All the things I think most people think about when they think of carbs. Corn, right? Rice, things like that. But what people don't think about very often is this. That's soda. That has a carbohydrate in it too. Which is a good point because of what we've been talking about, you and I, has been like dietary discussion, right? We're thinking diets and food, which is okay because that's what carbohydrates are. But now you need to get your brain into thinking about biology for a second. And remember that what we're about to talk about is slightly different than what you may have learned growing up. Because we're not just talking about diet, we're talking about what's in your cells. So carbohydrates, this big blanket term, they include sugars and polymers of sugars, which is what we have right here. So the sugar in that Coca-Cola, I get paid for this. I'm just kidding, I wish I did. It's not even my picture, I think it came from your textbook. But anyway, the sugar in that Coca-Cola, that's a carbohydrate. And then if you were to put those molecules, the monomers that make up that sugar and that soda, if you were to string them together, those same exact monomers, then you get what's in the pasta. So again, carbohydrates include the sugar, and then the polymers that are made from the sugars. Of course, if you're writing this down, you don't need to write anything about those 2 bullet points here down. It's just an example. Yes, there's small sugar molecules in soft drinks. Yes, there's large starch molecules in spaghetti and bread. But we'll talk about those when we get there because we're going to talk about each of these individually. We're going to talk about these small sugars and these large starches. But are there any questions about this slide? If there's anything you should know at this very point in time, it's that we are about to talk about carbohydrates. That's it, right? That's all you should know. Here's a little bit of information you also need to know. Where do you find them? What do they do? Obviously, there's all kinds of different creatures on Earth, but your book only talks about animals and plants. So we're not worried about fungi or bacteria, right? Just looking at animals and plants. So in animals, carbohydrates are the primary source of dietary energy. And it is also the raw material building other molecules. Which makes sense. And all of our molecules are carbon, right? Or not all of them, but as we learned earlier, most of them. And carbohydrates are also carbon-based. It's carbon skeleton. So we break them down to release energy and to build other molecules. That's what it is used for in animals. Any guesses what it's used for in plants? Basically the same thing, but also building material for much of the plant body. And you're going to learn there's a word for this. I'll go ahead and tell you, but no need to right now. We'll talk about it later in the chapter. It's cellulose. When you look at a tree, well, I mean, you see a bunch of green because that's chlorophyll. But most of the structure of that tree, it's made of cellulose. It's made of a carbohydrate. And you're going to learn about that later, so again, no need to write that down. Anyway, any questions about what carbohydrates do in animals or plants? If you're curious, for independent work, you could look it up for other things. What about protists? What about fungi? What about bacteria? What do they do? What do carbohydrates do for those living organisms? Anyway, let's move forward. Like I already said, carbohydrates. It's a big blanket term that includes 3 different types. Well, more than 3, but 3 that we're going to talk about. Monosaccharides, disaccharides, and polysaccharides. Here we go. The first one we're going to talk about is monosaccharides. Does anybody know what the prefix mono means? What does that indicate to you? 1. Good. Perfect. Yes, 1. That's a good start. That's actually probably the most important thing you need to know about monosaccharides. Those are the monomers. Like I told you earlier when I presented those terms, monomer, polymer, told you you'd have to know these things. So there's a test question for you right there. What is the monomer of a carbohydrate called? And the answer is monosaccharide. So for each of these, it's just the monomer, right? It's the one sugar molecule is a monosaccharide. Your book gives some examples. I'll talk about them. The questions might even be on the study guide. Don't put it next to it, because again, it's just an example. So I'm probably not going to ask you it on the exam. But some examples of monosaccharides are like what you might find in Gatorade. The thing that makes it sweet is different than this, than like table sugar. Table sugar is not a monosaccharide. We'll get to that later. But yeah, monosaccharides can be found in sugar, fruit, honey. Again, just examples you don't need to know. For this slide, the most important thing you need to know is the monosaccharides are the monomers of carbohydrates. You need to know that. So monosaccharides are not the monomers of proteins. They are not the monomers of DNA or nucleic acids. They are the monomers of carbohydrates. You need to know that. All right. This next picture I'm going to show you is going to show you what glucose looks like and what fructose looks like. Different monomers. There's no questions about this slide. You do not need to know this either. Again, this is just an example. But this is what glucose looks like when you draw it. This is what fructose looks like when you draw it. Looks pretty similar, right? It's got the same formula. C6H12O6, meaning there's 6 carbons, 12 hydrogens, 6 oxygens. Both of them have the same formula, but they have a different structure, slightly. And again, not being chemists, but knowing this relationship to structure and function, you should look at this and think, okay, well, they have a very similar structure that's slightly different. And then you can just go ahead and assume correctly that they also have a very similar function, just slightly different. Because, not that you need to know this, but fructose is slightly sweeter than glucose. All because of this right here, just because of where that carbon double bonded to the oxygen happens to be on the carbon scale. That makes it sweeter. And these, this is something you do need to know. When you have something that has the same formula but different arrangements, it's called an isomer. You need to know that. An isomer, same chemical formula, it's got a different structure, right? Different arrangement. How do you want to say it? Any questions about this slide? So again, the glucose and the fructose, those are just examples. For sure, I'm not going to show you a picture of these 2 things on the exam and say which one's glucose and which one's fructose. You don't need to know that. It's just a really great example, in my opinion, that again, similar structures but not exactly the same. Therefore, similar functions but not exactly the same. Also, notice too, imagine if you were to use your imagination, this carbon's here for this one, it's here for this one, so you could look it up if you're really bored for independent work. What if it was here? Then what would that be called? What if it was here? Then what would that be called? If it was here, that would be the same as that, but we won't get into that. So anyway, if you're interested, you can look that up. What are some of the other isomers of glucose, fructose, or basically C6H12O6? This next slide is everything we just said, that it's in writing for you. Glucose and fructose are isomers. So again, the fact that glucose and fructose are isomers isn't important. What is important is you know what isomers are, which are molecules that have the same molecular formula but different structures. So again, you don't need to know that glucose and fructose are isomers of each other. You just need to know what an isomer is, not the example. Any questions about that? All right, here's an example that's not in your book. And I know you can read, but it's really small, so you might not be able to, which would be helpful. Probably no one knows what this is, but you might. Does anybody know what these molecules are? I mean, there's the chemical name up there, but anyway, do you know what they are by any chance? I'll give you a hint. The one on the left is the main ingredient in Vicks Vapor Rub. So if you've ever used that and had that mentholy smell, that's what that is. And you can see these things look very similar, right? Basically, this molecule is a mirror version of this one, right? If this was a mirror, if you were to flip it over, that's what you would have. There's a word for that, but we don't get into it in this class. But anyway, that other molecule is the main ingredient in meth, right? So here you have similar molecules, the same formula, almost even the same structure. They're just mirrors of each other. And one of them you put on your kids when they have, an upper respiratory thing going on. The other one you use to make meth. So not that you need to know that. But the point here is that's a really great example of icebergs and how sometimes just one small change in chemical structure make a big difference. Anyway, back to the important stuff that you do need to know. Monosaccharides, particularly glucose, are the main fuels for cellular work. You should know that. And if you were to shorthand that, you could put glucose equals main fuel for cells. Because that's really the only one we talk about. Once we start moving forward and we start talking about cellular work, which will be chapters 5, chapter 6, chapter 7. Once we get out of this chapter, we don't look back. And we only stick with the glucose. Glucose is the one we're concerned with as far as monosaccharides. There are other ones, so keep that in the back of your mind. Glucose gets all the fame. You'll be tired of hearing about it. Another thing, this next bullet point you don't necessarily need to know. But yeah, when you put glucose in water, remember I just showed you that picture of glucose, right? The drawing, that straight long line. But it's not always like that. Sometimes when you put it in water, it forms rings. And the only reason that's really important, again, you guys aren't chemists, but that's important because like I said, they're monomers, right? That's the monomer. And you string a bunch of them together to make a polymer. Well, that's how they get strung together. It's the rings that get strung together. It's not these long molecules right here. So, any questions about that? All right. Next thing we're going to talk about. Wait, let's see if anybody can guess what we're going to talk about next. We just finished talking about monosaccharides. And I know I've already said it, but I didn't expect anybody to memorize it. What do you think the next one might be? We talk about monosaccharides. What might we talk about next? Logically. What comes after 1? 2. Good. So disaccharides is what we'll talk about next. For now, all you need to know that disaccharide, as you might imagine, instead of being 1 sugar, is 2 sugars. It's in the name, right? It is a double sugar constructed from, go figure, 2 monosaccharides. Because like we already said earlier, the monosaccharides are the monomer of carbohydrates. So if we're going to build something, using a dehydration reaction, by the way, we're going to use 2 monosaccharides. Now there's a bunch of different monosaccharides. So when you put different monosaccharides together, you get different disaccharides. You don't need to write this down, but these are some good examples. Sucrose, right? So table sugar. If you put together glucose and fructose, table sugar. If you put together galactose and glucose, you get lactose, which is milk sugar. If you put 2 glucoses together, you get maltose, something you might find in beer, malt candy. No need to write any of that down. The point is, obviously, if we have different monomers, different monosaccharides, and if you put them together in different combinations, you're going to end up with different types of disaccharides. Any questions about what's on this slide? The next slide, if you were to download the PowerPoint, it's an animation, so you can watch it. It shows you glucose and fructose coming together in a dehydration reaction to make sucrose, which is table sugar. Here's a drawing of a glucose and a galactose coming together to make lactose, again, milk sugar. That's what it's made up from, right? Put the glucose together with the galactose using a dehydration reaction. There you go. What you don't need to know is what's on this slide. Just some examples. Lactose in milk, maltose in beer, malted milkshakes, malted milk ball candy, and sucrose and table sugar. And we'll end it there. You didn't come in late, but you came in after I already got started, so if you don't mind sending me an email, tell me that you were on time, so I can already get it present. Anybody have questions? Let's say, I guess the last word for attendance is going to be beer. Just got finished talking about it. I will see you on WVSU Wednesday.